It seems to be also to be able to translate it from, let's say, preclinical models to more human-related models, which was really nice to see.
Fellow Homo sapiens, welcome to or welcome back to Epilepsy Sparks Insights. Now, I don't know about you, but although epilepsy genetics and the new discoveries enthrall me, the details and the terminology can get rather overwhelming at times. So I really do appreciate it when we have a PPI scientist who can speak regular lingo. So our guest this week is the winner of the Education and Public Engagement Champion of 2024 award, a future neuro, and today will share with us her microRNA discoveries. May I introduce to you postdoc researcher Mona Heiland.
Hello, my name is Mona. I'm a postdoc in David Henshaw's lab in the Royal College of Surgeons here in Dublin, in Ireland. And my work is on microRNA-based therapies for epilepsy, for especially drug-resistant epilepsies.
And as a neuroscientist, what type of epilepsies are you focused on?
Obviously, drug-resistant epilepsies, which means these are epilepsies types which don't respond to the current anti-seizure medications. And that can be all kinds of epilepsies, from common temporal epilepsies, which are one of the most common forms of acquired epilepsies. But I'm also interested in genetic form of epilepsies, mainly the ones which are caused by mutation in sodium channels.
Just for people who aren't familiar, could you give us an overview of sodium channels? And because lots of us, I reckon, and certainly me included, if you say sodium, I just think of salt.
It has nothing to do about the salt which you put in our food, even if it's also sodium included in there. So sodium ions are very important in the communication of neurons, so our cells in the brain. So how do neurons communicate? So there's one neuron which gets information from the outside world, and obviously it's so excited about it, it wants to give the information to all the other neurons in the brain. So they have to communicate somehow, and they do this by electrical signaling. So we have the sodium ions. So we have small, let's say, I call them doors or gates in our neurons. And they are not open all the time. They're like, you need to imagine these automatic doors, you know, when you enter buildings and you approach them and then they open. And then once you go through, they close again. They're a little bit like that. So they're sensitive to changes of electricity in the neuron. So if they sense a change in electricity, they will open up. And then all the little sodium ions that can go inside the cell. And this causes then basically a change in the voltage, and they are all spread over the whole length of the axon. So the axon is, let's say, the telephone line of a neuron to the next neuron, and they open, and the signal can run along the axon to the next neuron. They are not open all the time. After a while, they close again. Otherwise, there would constantly be electricity going on. And there are other channels there which are responsible to basically balance out the sodium signal. So these are done by potassium. So sodium and potassium, they are very important for our communication in the brain. And let's say if there are mistakes in the protein of which form the sodium channels, let's say in a door, if the handle is missing or it doesn't close properly, these are basically mutations in the sodium channels, then we get problems with the signaling. So if they're open too long, you get a lot of signaling. If they're not open enough, then you can get less signaling, and this is the case in epilepsy. So we have different types of sodium channels. Probably everyone heard about SCN1A, which causes Dravet syndrome. So Dravet syndrome has a mutation in the SCN1A channel, and this channel is not functioning, so it produces less signal. And then we have, on the other hand, something called SCN2A. So SCN2A, if it's basically too long open, it can also cause seizures. And the interesting part is different sodium channels are located in different neuron types. obviously our brain has different neuron types, so some are very chatty, they want to talk all the time, but imagine a room of people where everyone talks at the same time, it's just chaotic. So we need also someone who is coordinating that, and this we are called interneurons. And there, for example, in Dravet syndrome, the problem is SCN1A is an interneuron, so they have basically disruption in the coordination. So the interneurons that don't have enough signals, that's why all the other cells, they're chatting too much, and that's why it causes seizures.
Chatting too much, guys. That's a very polite way to put it, isn't it?
I tried to put it in a very nice way to say, well, basically it's chaos. In epilepsy, it's chaos in the brain because all the neurons, they fire at the same time, and the brain is just overwhelmed with it.
Tell me about your research, and particularly you've had a paper published about this work that you've done.
So I'm working on MicroRNAs. You did recently this beautiful podcast with David Henschel, who already introduced us very well into the world of MicroRNAs, also with his new book, which I really recommend to read because it gives the whole story of MicroRNAs. So in my work, I focus on a particular microRNA. So MicroRNAs, they have numbers for basically that's how they get identified, not like genes with name. MicroRNAs have numbers, so I'm working on microRNA 335. What I found is this microRNA is able to modulate the expression of voltage-gated sodium channels. So basically how it works, if microRNAs, as you heard in your previous podcast, they can basically regulate how much protein is produced by regulating gene expression. So basically they keep a balance in our body that we're not overflowing with some protein. However, if a microRNA level gets out of balance, like say if you have too much or too less, the protein has also changed. So what I found is if there are not enough levels of microRNA 365 existing, sodium channel levels go up. So we have way more sodium channel expression in the neurons. So basically they fire more, they get more excited. Basically you can get an epileptic phenotype. And then what we did the opposite is like we increased the levels of microRNA 365 and we could see the opposite. Basically, we could reduce down seizure severity and basically reduce down the sodium channels. So basically it goes into anti-epileptic properties. So by modulating this one microRNA, we could basically modulate the brain excitability.
And how did you find this out? Is this through working with rodents in the lab?
So how we found this microRNA is very interesting. It goes all the way back to the origin where we did our first podcast about cannabidiol CBD. So I looked originally which microRNAs are changed after treatment of CBD because I wanted to know if microRNAs are involved in the anti-seizure properties of CBD. And one of the microRNAs which was changed was microRNA 335.
Okay.
And then we found in blood plasma from temporal epilepsy patient, microRNA 335 is upregulated. And in the brain, it's downregulated. So it's differently in epilepsy, the levels of microRNA 335 are disrupted. And we found this also in another epilepsy model. And it's very nice that this one microRNA comes up in different contexts, like in human plasma and brain tissue, in a rodent model of epilepsy, and also through a treatment with CBD. Because CBD works very well in Dravet syndrome, we thought, well, maybe because 335 can modulate sodium channel, maybe it could be a potential way how CBD works in Dravet syndrome. It's probably not the only solution why it could work in Dravet syndrome. Probably it's a lot of things together, but we just thought we follow up what microRNA 335 is doing in general, and we found that it's really targeting sodium channels. Interesting is also we tried it in iPSC cells. So iPSC cells are stem cells from humans. So it's kind of a human model in the lab. And also what we saw there, that even in these human cells, if we block microRNA 335, sodium channel levels were increased. and they were more excitable. So it seems to be also to be able to translate it from, let's say, preclinical models to more human-related models, which was really nice to see. So what our plan is basically, where we are still in very early phase, but it would be cool to basically take our microRNA and only go into neurons where SCN1A is. And then we block this microRNA there and basically could potentially increase sodium currents to SCN1A.
Certainly keep your eye on Mona, because this is flipping exciting. You've got to figure out if it's, going to work on certain organisms before you do it in humans, right? But fingers crossed.
We have the possibility to get resected brain tissue from epilepsy patients and do tests on those ones. And this is the closest we get to humans at the moment. The translation is still very early phase, so we still need to find a way how to get it really later in a human being.
If you have a resection at any point, it's not necessarily the most ideal thing, or it certainly can be, but rest assured that at least sometimes your brain tissue removed could be used in research to help other people.
Exactly. We are very appreciated that some people donate their tissue.
Thank you so much to Mona for explaining her work in a way that those who aren't geneticists or neuroscientists can understand and enjoy. Do check out more about Mona and her work on the website torierobinson.com, where you can also access the podcast, the video and the transcription of this episode. And if you haven't already, don't forget to like, comment and subscribe to the channel, share this episode with your friends, colleagues, family members, whoever it might be, and see you next week.